Alright guys, today we're going to talk about the protists on the microbes and agents list on the micromission rules. So we're just going to go over the life cycles and the various functions of the specific species on the list. So general tip for the microbes and agents list, definitely be familiar with all of them and know how to visually identify them. So if they show you a picture, you should be able to tell them which organism is in the picture. And then they have a lot of complex things like complex life cycles, little details. So you don't have to remember every single detail, but just put all this information on your cheat sheet and then just have some like general background knowledge about each organism. So yeah, let's start with Plasmodium falciparum. So this is a protozoan parasite. So remember, protozoan means that it's a heterotroph, so it eats other organisms to get energy. And this parasite is the one that causes malaria in humans. So here, what we call the intermediate host, where the parasite is going to develop, is the human, while the definitive host, this is where the parasite is going to reproduce, is the Anopheles female mosquito. And this parasite is going to invade and infect our red blood cells, so erythrocytes. So when you see pictures of this parasite, it'll always be in a slide with all these red blood cells here. You can see these little crescent-shaped guys are the Plasmodium parasites. Perfect. So they're part of a phylum. So a phylum just means a classification of organisms. So it's kind of similar, like, family, genus. It's like one of those things. So phylum is called Apicomplexa, and this includes single-celled protozoan and obligate intracellular parasites, which means that these parasites can only survive when they're infecting a host. Like, they can't survive outside of the host. And then these apicomplexans, they're characterized by what's called an apical complex, which is the structure at the tip of these parasite cells. And it has this like complex here, and it secretes enzymes that allow these guys to attach to host cells, and it helps them invade and kind of wriggle their way inside of host cells. Then another feature that apicomplexans have is this apicoplast. It's a plastid, which means that it's like a chloroplast. It's related to chloroplast. It's from endosymbiosis, like we talked about in the last lecture. And it's evolved over time to become non-photosynthetic. So, and vestigial just means it used to do photosynthesis, but it can't anymore. And it has 4 membranes, which, if you remember from the last lecture, it means that it's undergone several endosymbiotic events in succession. So these plastids, we know they don't perform photosynthesis, but they definitely have essential roles for these parasites, because whenever we use drugs or remove these apicoplasts completely from the cell, these parasites will die. So we don't know exactly what they do, but we know they're essential for the survival of the apicomplexans. And then here's just some symptoms of malaria, because sometimes they'll give you a case study. So they'll give you like this description of a patient, and they'll give you all the symptoms and then the exposures, where they've been, etc. And then they'll ask you which of the microbes or agents is responsible for the disease. So here, malaria, the symptoms are fever, chills, headache, muscle aches, fatigue, nausea, vomiting, diarrhea, etc., etc. And the high-risk areas are in tropical regions, so like sub-Saharan Africa, Southeast Asia, and Central South America, basically where a lot of mosquitoes are, right? So let's look at the life cycle in detail here. So we're going to start with a mosquito that has the parasite, a mosquito that's already infected by the parasite. So the parasite will actually concentrate in the salivary glands in the mouth of the mosquito. So when this mosquito is infected and then it bites a human, it'll transfer those parasites into the blood of the human as it's drinking the blood. So it's going to inject what are called sporozoites. So sporozoites are just an early kind of immature, underdeveloped form of the parasite. And these guys will make their way through our bloodstream and then migrate to the liver cells of humans, called hepatocytes. So they'll kind of embed themselves here. Now, these sporozoites, they're going to mature, they're going to grow, they're going to get bigger. And eventually they'll undergo what's called schizogony. So schizogony is where, think of like mitosis, where the nucleus divides and then the cells are, you make a bunch of new cells, right? But in mitosis, we have one cell creating two identical cells, whereas in schizogony, we actually create a ton of cells. So like, for example, 20, for these parasites, it's like 20 to 30. So they'll form these, 20, 30 copies of themselves, and then they'll form these multinucleate, so cells with like many nuclei, and they're called schizonts here. So these schizonts, these schizonts contain many, many nuclei, and eventually they'll undergo schizogony to split up into like 20, 30 daughter cells. So these schizonts, like I said, they eventually rupture, and they're going to release what are called merozoites, which is the next stage of the virus. So these merozoites, they're going to swim through the bloodstream. And they're going to go, now they're going to exit the liver and go to our red blood cells in the bloodstream. So these guys will, using the apical complex that we talked about, they'll invade these red blood cells. They'll insert themselves inside, then they'll mature while they grow, and then they'll grow inside the red blood cells. So if you see pictures of Plasmodium, like on a microscope slide, often you'll see red blood cells with these like purple things embedded inside them. So these are the merozoites, and then those merozoites further develop into what are called trophozoites. So these trophozoites, they're motile, they can move around, and they're the active form of the parasite. So these guys are actively consuming nutrients from the red blood cell. And we call this early stage when the merozoite is growing into a trophozoite. We call this the ring stage because it forms this interesting ring structure that you can actually see under the microscope. These trophozoites, they'll grow bigger and bigger and bigger. Eventually, they'll undergo a schizogony like they did, like these sporozoites did before. So they'll undergo schizogony, form all these little new cells, new merozoites. Then that schizont will rupture like it did before and then release new merozoites. So these merozoites can now travel through the bloodstream and then infect more red blood cells, and then this cycle will repeat over and over and over like this. And I think I actually wrote it incorrect in the text here, but it should be that merozoites, either they grow into rings and then trophozoites and then go through this cycle, or they can actually, a rare few of them can turn into what are called gametocytes here. So these gametocytes are the sexual cells of the parasite. So think of like the sperm and the egg for humans. This is the equivalent for the parasite. So a few of these merozoites will turn into quote unquote sperm and egg. Then we'll see what happens from there. So, and then this is all takes place in the blood. Now, so the gametocytes can either be macrogametocytes, which are quote unquote female, and then microgametocytes, which are quote unquote male. So it's like the sperm and the egg, right? And these guys are going to get picked up by, since they're floating around the blood, a mosquito will come along, it'll suck up human blood that's infected, and it'll take these gametocytes with it, okay? So now these gametocytes, now they're inside the mosquito. So they come together, they fuse, they form the zygote, which is like basically like the new embryo of the Plasmodium. Think of it that way. So then the zygote, it'll turn into what's called an ookinete. These ookinete, I'm not sure how to say it. So and these guys move around, they're relocating inside the mosquito, and then they further develop into what's called an oocyst. So you can see that on the left here, ookinete, and then oocyst. These oocysts are, they form this protective cell wall around them, and it's very like a rigid structure that like houses this new growing parasite. Okay. So within the oocyst, the parasite's going to replicate many times. Eventually, it's going to rupture. So you have this bursting cyst here, and it's going to release a ton of new sporozoites, which are now going to go to the salivary glands of the mosquito. And now this mosquito is carrying the sporozoites in its salivary glands. It goes and bites a new human, transfers those sporozoites, and then we start this cycle all over again. So as you can see, the life cycle is pretty complicated. The exact definitions of all these terms like ookinete, oocyst, etc., not super important, but just know, at least be familiar with these terms and know the context that they fit in. And then you can put one of these charts like on your cheat sheet or something, and then so that you can reference whenever you need to answer questions about these. And then interesting part of the life cycle, sometimes the Plasmodium falciparum, they form hypnozoites when they're inside the liver. So these hypnozoites, they're this dominant resistant form of the parasite that kind of just sits there for months or even years. So you might get malaria first, and then you come back, now you're healed, but these hypnozoites might still be hiding around in your liver, and then months or years later, they can reactivate and then basically come out of dormancy and then start infecting you with malaria again. So it's kind of a dirty trick that this parasite has. So let's watch this short video that'll give you a actual animation so that this will make more sense to you guys. Here we go.
The malaria parasite's life cycle is very complex, involving many stages. As the mosquito feeds, malaria sporozoites enter into the human bloodstream. They quickly make their way to the liver and infect liver cells with the help of the circumsporozoite protein. The parasite develops into mature schizons, creating tens of thousands of merozoites, which burst out of the cell and journey back toward the bloodstream. They then infect healthy red blood cells, reproducing and eventually rupturing the cell, liberating more merozoites, which in turn go on to infect other cells, leading to the massive destruction of red blood cells. A few of the infected cells develop into gametocytes, which can remain in the bloodstream for several days and may be ingested by another mosquito during the subsequent feeding. Inside the mosquito, the gametocytes develop into new sporozoites. So now the human has infected the mosquito. The life cycle comes full circle and continues.
Awesome. So there, hopefully that makes a little more sense. Now talking about protective measures for malaria. Chloroquine, this was the drug of choice for a few decades, but recently it's become, Plasmodium has become pretty resistant, so they're not really using this anymore. And now the most commonly used therapy is called artemisinin-based combination therapy, or ACT. And this will prevent the early, so once the merozoite infects the cell and then it goes through the ring stage where it's growing into a trophozoite, it'll stop the life cycle there. So then other drug treatments, they include, we have quinine, which interferes with the digestion that the parasite does when it's like eating up our nutrients in the red blood cell. And then we also have, you will pair quinine with doxycycline or clindamycin. And these are antibiotics that have, I don't know the exact role that they have, but for the parasite, but they're effective antibiotics that I guess have some related role that helps kill the parasite as well. And a lot of these drugs actually, we don't know exactly what the mechanism is by that they interfere with the parasite's life cycle. We don't know the exact mechanism, but we know that when we use these drugs, these parasites die. So just interesting thing to know about like pharmacology in general. We don't always know exactly what the drug's doing, but we know that it's effective. So then we have another measure is called, we use IV, so IV injection, intravenous injection. And we use artesunate, which is derived from this compound that's in ACT. There's artemisinin. And this is for especially severe cases because we're directly injecting the drug into our blood. Okay, now let's move on to Nanochloropsis species. So when you see this SP here, it just means there's this group called Nanochloropsis, and then there's a bunch of species that fall under this group. So this one includes 6 species of microalgae that are unicellular and usually found in salt water. So microalgae, if you see that green stuff growing on the surface of ponds, like that's all the algae, microalgae. So a lot of times it's used as a food source, especially for like fish, smaller organisms, and then also sometimes livestock, because you can kind of dry up all these cells and make them into a powder. And then you can feed them to various animals, and they have good nutrient profile. And the nutrients are so good that even sometimes we use them as human dietary supplements because they contain fatty acids. So like if you've heard of like omega-3 fatty acid supplements, stuff like that. Then they also have carotenoids, which include some like vitamins that are important for our body. Like I believe vitamin A falls under that category. So these microalgae, they're pretty useful for nutrients. Recently, they've also been used for what's called algal biofuel production. So algal biofuels, they're basically you can grow algae and then you can extract some of the products that they create naturally, and then you can purify those products and then you can use them as fuel. So it's a renewable energy source that could potentially replace some of the more pollutive, wasteful energy sources that we have today, like fossil fuels, petroleum, etc. And it's also renewable because like these algae will continue to create these energy sources like throughout as long as they're alive. So these nanochloropsis, they have specific energy-rich lipids that we can harvest and then use and purify into biofuels. So the general process for doing algal biofuels, making these, getting them ready, involves, so we have harvesting, dewatering, extraction, processing. So harvesting is basically where after we grow these cells in these big vats, we got to get all the cells out. So we have a variety of ways. These, you don't have to really know what these are, but I'll go through them. Flocculation. This is where you add a chemical that helps all the algae cells stick together so that you can like filter them out easier. Filtration, just filtration. Then centrifugation, this is where you'll spin the cells really, really fast so that basically all the cells will collect at the bottom and then all the liquid will be on the top. So you can just get rid of the liquid and then you'll have all the cells at the bottom. Then next, you got to dewater or dry. So you got to get all the moisture out of these cells, right? Then a third, extraction. Now we got to extract the product that we want. So these energy-rich lipids, we're getting them out of the cells. We're taking them out of the cells. Then we're for processing. So we're doing some chemical reactions with enzymes and then other chemicals to create the final purified oil product that we want to use as our fuel. So I think on the Oklahoma test, a lot of times they put like harvesting, dewatering, extraction, processing, and then talk about biofuels. So definitely know these 4 steps. You don't have to know exactly what happens in each step, but if you're curious, I did leave a link here that you can read about in more specificity. That's from the OSU Agricultural Science Department, I believe. So. Yeah, so we grow these guys in these like either these big open raceway ponds or photobioreactors, and these are just like little tubes that we can control the environmental conditions in and then extract all the stuff from there. So and then some other details, nanochloropsis. So it produces more lipids, more potential energy products when they're grown under conditions where there's not that much nitrogen, and then they can also, which is very interesting, they can be genetically engineered. We can literally change the DNA of these microalgae so that they produce more lipids and more potential energy products, as well as some other factors. So the advantage of using algal biofuels is that, first of all, obviously they're very good for the environment. We're not using fossil fuels. And then also land usage is minimal. So it's a lot less stress on the environment. And then microalgae, they're pretty easy to grow and they replicate fast. So we can make a ton of these really fast. So the biofuel production, at least growing the cells will be will happen in a timely manner. And then, like I said before, these strains it just means like kind of like species or types or microalgae. They can you can genetically modify them so that they produce more of the product that you want from them. And so the limitations are that algal biofuels, they're actually much more expensive than petroleum, standard oil. So no one, people aren't buying algal biofuel because it's way more expensive than petroleum. So there's no point, right? So, and then so on the whole price thing, so extraction and harvesting, these processes, they can be convoluted and also have high production costs. And then large scale growth also requires a lot of water since you got to, culture the algae inside water. All right, let's move on to paramecium species. So paramecium, again, a group of organisms under which there are multiple species included in this group. So these guys are, they're common freshwater unicellular heterotrophic protists or protozoans. And they're classified as ciliates, which means they have many, many cilia, little hair-like projections on the outside of the cell that help them swim through the water. And these actually have pretty big cells, so they can be seen by the naked eye, 50 to 300 micrometers here, microns. And these are actually pretty cool because you can find them in pretty much any lake or a pond. So even in Mr. Del Grosso's class last year, we actually like we got some lake water, from the Cassidy Lake, and then we looked at drops under the microscope, and then we can actually see some paramecia. So they're pretty ubiquitous, pretty common. All right, so let's look at the reproduction of these guys, their life cycle. So recall from the last lecture where I mentioned briefly that paramecia have two nuclei. So they have this micronucleus that's really small, and they can actually have multiple of these, but the micronucleus is polyploid, meaning it has many copies of the DNA. There's many copies of it, and it also it contains the genes that are going to be passed onto the offspring, so the next generation. And then the macronucleus, it contains the genes for everything else that the cell needs to do, and it is actually derived from the micronucleus. Yeah, so these paramecia, normally they're going to just reproduce asexually through what we call binary fission. So if you haven't heard of this term, it just means, think like mitosis in human cells. So you start with the organism on its own, it replicates its DNA, then it splits in half, so it's creating two identical offspring from one cell that it started with. There's not a lot of genetic variation, right? It's just like almost like cloning yourself and making another copy of yourself. But they have two nuclei, which makes the process slightly different from other organisms. Two or more nuclei. So the micronucleus is going to divide mitotically, meaning the spindle fibers are involved, which means, if you remember, you had the mitotic spindle, you had these microtubules coming in and grabbing those chromosomes, right, and anaphase and separating them out. So the micronucleus is separating like that, and then the macronucleus is dividing amitotically, which means it doesn't have these spindles. Instead, it just starts out here, and then it literally physically pinches off and then separates out like this into two nuclei. Then the cells will just divide, and then, yeah. All right, so I think I forgot to put a picture here, but that's fine. Some paramecia, they also do what's called conjugation. So this is between different organisms. Two paramecia can actually transfer their DNA. So this is going to increase the genetic diversity significantly, right? Because paramecia, normally they reproduce asexually, which means they just make clones of themselves. So if you recall from biology, having low genetic diversity is not favorable, because if the entire population is exactly the same and exactly the samely adapted to the same conditions, then let's say the environment changes drastically. Now all of the population is just going to die out, right? Because they're not adapted to it. But if you have genetic variation, even if the environment changes a lot, like some of the cells will probably be well adapted to that environmental change, so they can survive, and then the species can continue to grow. So that's why this conjugation is important. And also, after a cell, so after a paramecium, undergoes a few hundred rounds of normal asexual reproduction, so binary fission, so it's making copies of itself over and over and over again. You can imagine every time it makes a copy, there might be a few mistakes in the DNA. So if you make 100 divisions, the cell at the very end of that is probably going to have a decent amount of like messed up DNA or like mistakes when it was copying that DNA, right? So in the lab, you notice that when cells have undergone a few hundred rounds, they actually become kind of shriveled up. They don't move as well, and they're like kind of static. So they're not fully functioning. So basically, when you perform conjugation, one of those like more dead paramecia can conjugate with a normal paramecia, and then that gene exchange actually rejuvenates the cells. So the cell is now more active because it got some DNA transfer from a paramecium that's like normal, pretty much. So there's another reason why these guys do this. And then conjugation, it's actually most well known in bacteria, but we'll discuss that in a future lecture. So we'll start off by talking about this guy here. So paramecia, some of them have two mating types, which are called O and E. So it's like male and female, but then we have O and E, odd and even. So let's see how conjugation occurs. So first we're going to start by fusing what we call the oral grooves of the two paramecia of opposite mating types. So let's say we have an O and an E here. What happens is that, so the oral groove, if you recall, if this is the cell, is this mouth-like area where all the food will get sucked into and then digested within the cell. So the oral grooves, and also called the cytostome, right? Cytostome. So these will fuse between two cells, form what's called the protoplasmic bridge. This just means like the connection between the two cytoplasms. So yeah, so next, the macronuclei, or the nuclei that contain the non-sexual genes, right? The big nuclei are degraded, and then eventually they just disintegrate. And then each micronucleus is now going to undergo meiosis, forming 4 haploid entities. This just means like we start with 1 micronucleus, then we get 4 micronuclei that have half the amount of genetic material that the first one had, like this. So it's just like meiosis when we had like forming gametes in humans, so sperm and egg, right? So it's the same process here, except it occurs with the micronuclei. So from there, we'll form 4 haploid entities, and 3 of the 4 will actually disintegrate and go away. So we're left with 1 micronucleus in each cell. And so here we go. So now we have 1 in each cell, and then the other 3 in each cell are gone. Then now this micronuclei will undergo division and form 2 gamete nuclei. So it'll undergo an asymmetric division, which we'll see on the next slide here. And these are called 2 gamete nuclei. So here you see the 2 gamete nuclei here. So like I said, it's an asymmetric division, which means that one is going to be bigger than the other. So we call the bigger one the stationary nucleus, and we call the smaller one the migratory nucleus. So what's going to happen is that the migratory nucleus of one cell will go to the stationary nucleus of the other cell. So this is where the genetic transfer actually occurs. So just write trans. So the transfer occurs during this step where each migratory nucleus will migrate to the other cell and then combine. And then once it combines, it'll form these zygotic nuclei here. So now each paramecium has 1 single nucleus. Now, after this happens, these guys will split apart and then go off on their ways. And then this next part is not that important, so I'm not going into detail, but essentially this 1 zygotic nuclei will divide multiple times and eventually turn into 4 resulting cells here. And then each of them will have a micro and a macro nucleus, just like the paramecia that we started with here, right? Okay, so some terminology when the 2 cells are together. When they're fused together, they're called conjugates, and then after they're separated, they're referred to as X-conjugates. So these 2 cells, so just overview, what happens is that the 2 cells will exchange genetic information. Now we have 2 new cells that are genetically mixed up, and then each one of these will turn into 4 new cells. So we'll end up with 8 total cells here, this. So once we have, 2 to 8 is the overall idea. So like I said, going from this step here, going from the 2 resulting to the 8 total, this step is very complicated, so I'm not going over that in detail, and I don't think you'll have to know it for the tests, but they're included in the next video. So if you're curious or interested, you can go ahead and look over those in the video that is in the next slide here. And I also left a link here that has more specific descriptions of each step if you're interested. And this is not required by any means. So yeah, I have 2 videos here, and I won't play them right now, but you guys can go and watch them on your own time. But hopefully this gives you a good basis for the protists on the list of microbes and agents.